we know P and Q, which represent what?
10.
No. Allele frequencies. Allele frequencies. P and Q are representative of allele frequencies, and they're just, you can use any letter you want, I don't care, really, but P and Q are sort of classics. So, know your P's and Q's. But if you know those allele frequencies, you also know what? Genotype, expected genotype frequencies, right? And if the population's in Hardy-Weinberg equilibrium, then if you know P and Q, you do know P squared, 2PQ, and Q squared. If the population is not in equilibrium, do you know that?
No.
But you always know P and Q because you can calculate allele frequencies from data sets. That's what you do. You can always calculate the allele frequencies. The idea of equilibrium is what you observe, what you're expecting to observe given the allele frequencies, right? That's the whole idea. Any questions on Hardy-Weinberg? Because we're going to use it to do a few things. But if we know P and the populations in Hardy-Weinberg equilibrium, or the locus, it doesn't have to be the whole population, but that locus is in Hardy-Weinberg equilibrium, then we also know P prime, the allele frequency in the next generation, right? Because it's going to be the same. It doesn't change. That's the whole idea of Hardy-Weinberg is it defines a population in which P and Q are known, which also defines all the genotype frequencies and tells you what the population is going to do for the rest of its life, which is nothing. It's going to sit there, those allele frequencies. But what if this is not true? What if we can't predict the next? That means what?
That's not in equilibrium.
Not in equilibrium, which
It's
means what happened?
Selection occurred, bro?
Maybe selection. Something's happening, right?
Yeah.
There's a long list of no's, right? I had.
Right.
It might have been migration. It could have been mutation. Might have been selection. Could have been drift, which you'll see, which is has to do with large population sizes. There could have been non-random mating as well. So it could be a whole number of things. So when your population is not in Hardy-Weinberg equilibrium, you lose the sort of ability to predict genotype frequencies and predict what the genotype or allele frequencies will be in the next generation. So let's start with violating one of those assumptions, and that is the assumption of no selection. Let's say there is selection, and you're all biology majors or somewhat related majors, and someone asks you, so what's this natural selection stuff? What do you tell them?
It's a mechanism.
It's a mechanism of evolution, that's important. What does it do? What's the ultimate outcome of it?
Selects for favorable traits to produce.
Selects for favorable traits, and what happens to those favorable traits in the population?
They get passed on as genetic
Passed
information.
on, but they get more frequent.
Right.
Right? So the population ultimately becomes adaptive. The whole idea of selection is adaptable or adaptive evolution, right? The ultimate goal of selection, which is really sort of just differential reproduction and survivorship, right? That leads to adaptation. Now, if I can sort of shorthand the whole bloody thing, right? Adaptive selection is there's books and books on it, but really it's differential reproduction survivorship among individuals that leads to adaptive evolution. The population becomes more fit to its environment. That is to say, in population genetics speak, because that's what we're doing, right? We're going to do population genetics. When we talk about fitness, we talk about this little, we usually abbreviate it little w. Don't ask me why. Fitness doesn't start with W. I know it doesn't even have a W. But this is the term for fitness in population genetics. Selection is always going to increase the average fitness of the population. No matter what else selection is adaptive evolution, which means if it's adaptive evolution, it's going to work to increase the average fitness of the population. Yes? So the population becomes sort of better at surviving and reproducing in whatever environment it happens to find itself in. Yes?
Mhm.
So that's average fitness, the W with the bar over it. I'm going to reach way back in the history of biology in 1981. So this is a rather sort of old experiment. I know, the 80s. It was a long time ago. Seems like only yesterday. But Kavner and Clegg worked with
It's weird.
that's hard to write in font. Drosophila, that famous of famous beasts from your computer screen that you worked with, right? Did you actually ever see Drosophila?
Yeah.
Real ones?
Yeah.
No.
What?
Well,
You must keep
that's
a pretty clean house, because they're always in my house come spring, summer.
No, I told us that those aren't, your house is just dirty if you see those flies or something.
Well,
Those
yeah,
aren't fruit
because
flies.
we
That's when you let bananas go right.
exactly. Where you leave a little fruit out, you get fruit flies.
Yeah. You Australia's really bad with them.
There's all kinds, so yeah. I mean, this is why they're worked with. They're easy to get. Like biologists are not sort of wealthy folks usually, so they work with things like flower beetles, Tribolium, another sort of famous one, Drosophila, nematode worms. These are the famous organisms of biology, right? Something you can go get out of anybody's garbage. Drosophila. They also work with something else, an enzyme. An enzyme locus called alcohol dehydrogenase. Maybe you're familiar with it. The alcohol dehydrogenase locus had two alleles, what they call ADH fast and ADH slow. Now, what does alcohol dehydrogenase do? It dehydrogenates alcohol?
I was gonna say this, hydrogen, and alcohol.
All right, I don't want to know the organic chemistry, but effectively, it's going to break down ethanol, right? And you know that when fruit flies live around fruit, sometimes ethanol is produced in
Mm-hmm.
fruit, wine and such. So they had two. The ADH fast locus broke down ethanol faster than the ADH slow locus, thus the name. Makes sense, right? Sometimes that happens. They constructed an experiment, of course, and on the y-axis we're going to show some data here. On the y-axis is going to be the frequency of the ADH fast allele, and then this is going to be time on the x-axis. And time, my friends, in evolution is measured in generations. Right? So, not time on your watch, not time on the calendar, not time by any other means, but generations. And they went, I believe, about 50 generations. So they started at time 0. They had, well, they had more than that. They had 2 treatments, generally speaking. One was a control treatment. The control treatment was Drosophila living in happy Drosophila land with lots of Drosophila food to eat and nothing to eat the Drosophila, and just rainbows and butterflies and all kinds of things. Very nice controlled environment. That was the controlled treatment, right? No pressure, just happy. They constructed the experiment so that the allele frequencies at the start of the experiment were 50-50. 0.5 and 0.5. So there were the allele frequency of ADHF was 0.5 and the allele frequency of ADHS was 0.5. Now, in this controlled experiment, what would you expect the frequency of the ADH fast allele to do over 50 generations?
Increase.
In a benign environment. Stay
Stay
relatively the same, right? Which is exactly what it does. It is relatively the same. This was the control. That's what you wanted the control to do anyway, right? So they also had a treatment where they had different levels of ethanol presented within the media that the fruit flies have to sort of deal with. And ethanol, as you may know, can be toxic at certain levels. Right? Hopefully you don't have experience with that. But ethanol can be toxic. So this presents sort of a selective pressure, a selective environment. You hear that in microbiology all the time. You're presenting selective environments, right, to the bacteria to help them to sort out something. So what do you think happens to the frequency of ADH fast in the experimental treatments?
Increase.
It increases, right? So this one has ethanol. Voila! Selection has occurred, right? Maybe. Surely, in the control, the genetic structure of the population hasn't really changed. It's very similar with respect to this locus, right? So the population effectively hasn't evolved much, if at all. But in the experimental treatments, we definitely have this sort of directed evolution where the ADH fast allele becomes more and more frequent in the population. Why? You have to process ethanol, and the better you are at processing ethanol, the more likely you're going to survive, and the more likely you're going to be successful in reproduction, right?
Mm-hmm.
So what happens to the frequency of that allele?
Increase.
That allele is directly tied to fitness. It increases. Get it? Easy, right? Yeah. So that's just a very sort of simple experiment of natural selection in action on a particular locus that's tied to a particular phenotype, that is a physiological phenotype of ethanol turnover in the body. But if we know something about selection and we know starting allele frequencies and things like that, can we actually predict how selection will change allele frequencies? Can we predict the slope and how quickly allele frequencies will change? Of course we can. It's really easy. So, in evolutionary biology, in population genetics, we're always interested in how populations are changing genetically. These selection models allow us to sort of predict exactly how populations will change, given certain selection pressures and so forth. So we can predict, if we know P, and we know something about selection going on in the population, then we can predict P prime. In order to do that, we need first, there's an algorithm of two equations that we'll need to iterate. And I'll give you an example of how it works, but let me give you the equations first. And they're rooted in Hardy-Weinberg's equilibrium, but weighted by differences in fitness between genotypes. So first thing we need to do is calculate the average fitness of the population to begin with. The equation for that is P squared, the homozygous genotype, times the fitness of that particular genotype, plus 2pq, times the fitness of that genotype, plus q squared, times the fitness of that genotype. So the fitness is p squared, and then multiplied by what I call W11, which is the fitness of the p squared genotype, the homozygote, right? The 2pq times the fitness of the heterozygote, which I labeled W sub 1, 2. And then the fitness of the other homozygote, q squared, times W22. Okay? You see that? So it's just Hardy-Weinberg's little scheme, but this is a weighted average. It's weighted by the relative fitness. Relative fitness is a contribution of that genotype to the next generation, effectively, right? How well they survive and reproduce. So this little W becomes a measure of how much this one's contributing to the next generation. Once we have average fitness, we can then calculate P in the next generation, that's P prime, with the following equation. And that's P squared times W11 again, plus PQ, W12 times W12, divided by the average fitness. To read it another way, that's the homozygotes times their fitness, plus 1/2 the heterozygotes times their fitness. Remember how you get allele frequencies, homozygotes plus 1/2 the heterozygotes? That's p squared plus pq, not 2pq. I didn't write 2pq, did I? That's a common mistake, and I often do it myself, because I get in the habit of 2pq. Divided by the average fitness. What this will give you is allele frequency in the next generation. Okay? In order to sort of find out P in any generation, starting with a given sort of allele frequency and given a certain fitness regime, then you can iterate this as many times as you need to, a million, bajillions, whatever. What you have to do is do equation 1, then do equation 2, then do equation 1, then do equation 2. 2, 1, 2, 1, 2, right? The common mistake here is, what you'll see is what we get out of here is P prime, which means we also then know Q prime. And then you have to go back and recalculate a new average fitness, because what does selection do to the average fitness of the population?
Changes it.
Changes it. It's trying to increase it, right? Selection is going to increase the average fitness of the population, so every generation the fitness should be sort of creeping up. If you do this iteration and your fitness is going down, there's something wrong with your math. Okay? Selection increases average fitness. That's what it does. And to some point, it'll get to an asymptote where it can't go any higher. And you'll see that in a little bit. Does everybody understand those equations? So that we can do some real numbers with them. Real quick, so as not to bore you too much with this selection stuff. So, you'll see me do this a bunch tonight. There are 3 genotypes, because we're going to do everything with 2 alleles for now. Makes our lives simple. So we have 3 genotypes, and let's say we go out and measure how many offspring each genotype produces. And we find out that the A1 genotype produces 50 offspring, and the A1A2 produces 25, and this one doesn't produce any. Let's also sort of make an assumption that we're starting with allele frequencies of 0.5. Because that makes our lives easy too. So, in order to do this little equation over here and to find little w, that fitness we want, we can't really plug in these whole numbers of 50, 25, and 0. It won't work. We need to sort of standardize them. In other words, we need to, because fitness is a relative term, right? Remember that? Fitness is a relative term. Your fitness is relative to the other individuals in the population. So what we do in population genetics to get little w is say, here's the best genotype, right? It's producing the most offspring. With respect to the data we have, that's the best one. So then we say that becomes the denominator. So little w becomes 1. Everybody's fitness gets sort of divided by the best fitness. Yes? You see that? So that this would be 25 over 50. So the fitness of the heterozygote would be 0.5, and of course, that's still a 0. Right? So the most fit genotype gets a fitness of 1, and everybody else is sort of scaled accordingly to that. Got it? That allows us to use that little w in this equation, because remember, we're just dealing in fractions over here. We can't start slapping whole numbers in there. The numbers will blow up pretty bad. Got it? So what's the first thing we need to do? Calculate. So the average fitness is going to be p squared times the fitness of the A1 homozygote, because remember, this is effectively p squared, 2pq, and q squared, representing them, right? So that's going to be p squared 0.25 times 1, so it's going to be 0.25. 2pq is going to be 0.5, right? So 0.5 times 0.5 is 2.5 times 2, or 2.25, and then times 2 is 0.5, so 2pq. The heterozygote frequency is expected to be 50%, yes? With these allele frequencies, this is expected to be 0.5, and then we're going to multiply it by 0.5, because that's the fitness of that. Start over. Square, 2pq, W1, 2, squared, W2.
Okay, so W11 is the thickness of the homozygote? Okay, that's one, okay.
Yeah.
Okay, where'd you get the 2.25 from?
P squared. Here's P.
Oh,
Here. Sorry, I forgot to. Frequency of A1 is 0.5, which is P. Forgot to finish my definition there. See where they come from? Get it? If you sort of do all that out, you're going to get an average fitness of 0.5 for this particular population. Now we have what we need to calculate P in the next generation becomes it's just a matter of plugging in the numbers, but it's P squared times its fitness, which is 1, plus PQ, oops, I did the wrong PQ.
Yeah.
See, I did 2 PQ there. Don't do that. Sorry. See, it's so easy to make a simple mistake. If you sort of run those numbers out, you'll end up with 0.75 as the allele frequency of P prime, which means the frequency of A1 in the next generation is 0.75. So what's the frequency of A2 then? 0.25. Right? So what happened to A1? The frequency increased. Why? Look at the fitness. Which genotypes are contributing to the next generation? The A2 homozygote surely is not.
Yeah, this was the first.
This is an example of very, very strong selection, right? Most of the time it's not that strong. So allele frequencies will change, but not by that kind of leap. That's a giant leap. But it's just it's sort of a nice example. Does everybody see where that came from? If you wanted to do P in the next generation, you take this 0.75 and the 0.25, plug it into that again, go round and round again. The problems online ask you to do that for 3 generations, maybe like that. The easiest way to do that, you guys know something about Excel, right? You can tell Excel to sort of do this equation and then tell it to do it over and over and over and over and over and over as many times as you want. You can do it like a million times. Not like you want to, but on an exam, you'll probably ask to do it for at least 2 or 3 generations. But if you're in a pinch on the exam, use a little test-taking skill. Figure out the first one and see like, oh, it's moving a little bit, so if I sort of go that much each time, it changes each time, but still, first of all, your average fitness should be increasing, so you always make sure of that. And then if your A1 allele is apparently the sort of best fit allele because the genotypes associated with A1 are better fit, and A1 frequency is going down, then there's something wrong. So think about it sort of all together. Don't get tied up in the math so much. Your answer should make sense in the context of the data, right, that you've been given. And so that's, just check yourself. Don't fall into little traps. Because if you do 2PQ over here, you'll get a weird answer and it'll make things go the other way sometimes. So beware, calculate with care. The carpenter's rule to measure twice and cut once. Right? That's what you're supposed to do. I tell students in my lab, calculate twice, pipette once. It's expensive to mess up. So let's look at how selection can change allele frequencies with sort of different scenarios of allelic relationships. We've seen in the ethanol case, the ADHF allele goes up, and it goes up in a fairly linear way, very straight. But I want to do something a little bit different. So let's set up a scenario where we begin to introduce the idea of dominance and recessive relationships among alleles, and see how that sort of affects how allele frequencies will change. And then compare that to sort of a variety of situations, as it were. So, if A1 is dominant to A2, complete dominance, then we should have, and A1 is the most fit genotype, or fit allele, then we should have a selection or fitness scenario of this. So these are the little W's. The little W is 1, and because the heterozygote looks, because of the dominance, looks exactly like the homozygote. Remember, selection happens on the phenotype. And then the other homozygote gets this sort of little deflection. The little s is a selection coefficient. You've probably seen it before in other situations, right? Maybe little s. Selection coefficient. It's sort of the deflection from best fit. So in this case, whatever, and it doesn't really matter for our purposes what little s is. It's just telling you that that genotype is not as fit as the other two by some factor. But before we sort of go too far, I would like to sort of sort out what exactly dominance means. So here we have on the x-axis 3 genotypes, the same classic genotypes I've been using, A1, A1, and so on and so forth. On the y-axis is some trait value, whatever you want it to be. If you want to write something here for a trait value, that's fine. But a trait value. So in this case, just like I've said, A1 has a higher trait value than A2. Now, how do we sort of determine dominance and recessive? If these two alleles were co-dominant to each other, what would the heterozygote look like?
0.5.
0.5 meaning intermediate, right?
Right.
In other words, there is no dominance in that. Both alleles contribute equally to the trait, as it were. Yes? If A1 is completely dominant to A2, what should the trait value look like? Right? That's a situation of complete dominance, or we say D equals 1. If A2 is completely dominant to A1, the heterozygote would look like A2. Again, complete dominance. And D would equal, in this case, -1. Dominance, or what we call in quantitative genetics, the dominance deviation, is sort of how far off the heterozygote is from the exact intermediate between the homozygotes. So codominance means there's no dominance deviation. Complete dominance means one masks the other. That's what you're used to, right? The sort of idea of big A, little a, and one masks the other one. Now, so what's partial dominance then? It's all in here. Partial dominance is sort of not quite completely dominance, but some deflection from completely intermediate. Either down or up, doesn't matter, right? You've heard those terms before? Dominance is a relationship between alleles. That's what it's referring to. It's often sort of defined in genetics, if you're doing sort of trait stuff, as one masking another. Now what happens if the heterozygote is up here? What do you call that? Or down here below the trait value of the sort of lowest trait value. It's related to that. Overdominance. Overdominance, right. This is a situation where the heterozygote's trait value is above that of the highest trait value, overdominance. And then what would you guess this one is? Underdominance. Right. So we have a variety of situations.
I'm sorry, could you elaborate on what you mean by trait value again?
Could be anything. Could be like, you could just substitute any trait in there. Ear size. I mean ear of corn. Most of your corn out there is hybrid corn. If you look at the parents, they're only about yay big, but the ones you buy at the market are not. They hybridize to make a nice marketable product. It's an example of hybrid vigor or overdominance. So just any trait. But when the heterozygote is sort of got a trait value that's greater than the trait value of any of the other two homozygotes, then that's overdominance. Or if it's below, it's underdominance. It's sort of a bizarre relationship because it sort of falls outside of what you would expect, right? I mean, how can it be better? But then how could it be worse too? But it can be. It happens. All right, so that's how dominance sort of, and that's, the only reason I do that is not because I think you need a lesson on dominance, is because when we get to quantitative genetics, that sort of, we'll come back to that. We'll need that for that, so tuck that away. You know, don't just forget it after the next exam. You need it again. So here we have a situation of fitnesses where the A1 allele is dominant to the A2 allele. So what we want to do is sort of figure out how the population frequencies are going to change over time, given this particular relationship among alleles and fitness relationships among alleles. Keep in mind that dominance and recessive terminology says nothing about the actual fitness. Right? A recessive trait is not necessarily less fit than a dominant trait by any stretch. The idea of dominance and recessive is a relationship among alleles and really says nothing about the fitness of those things. So don't get it confused. Don't assume dominance is always more fit. Sometimes people do that, and it's not quite right. That's a trap. Watch out for it. So we have allele frequency on the y-axis and then time or generations on the x-axis. What I'm going to do is start the allele frequency of A1 down here at the bottom and A2 up here towards the top, just for fun. So this frequency of 1 at the top represents what we call in population genetics fixation. And the frequency of 0 is what we call extinction. Because if you think about it, if the frequency of A2 goes to 1, what happens to the frequency of A1? It went to 0. So we say one allele went to fixation, the other to extinction. Fixation is becoming a frequency of 1, which means there's no genetic variation left in the population because there's only one allele left, monomorphic. And if that's the case, that locus with one allele is no longer has any selection potential, right? Because there's no variation. And if one goes to fixation, the other must go to extinction because P plus Q has to equal 1. And if P equals 1, then Q has to equal 0. So what's going to happen to the frequency of A1 given that scenario? Up, down, round and round, how's it going to go? We can start and kind of down there in the left corner.
Up.
It's going to go up,
Go on.
right? Does everybody see why it would go up? A1, and the heterozygote, that's the best fit genotypes. So A1 is sort of in league with the best genotypes. So it is going to go up, and I'll tell you it is going to go to fixation after some point, so it is going to go eventually, if the selection plays out over time, no matter what that little s is, no matter what the selection coefficient against it, that's just going to sort of just determine how quickly it gets there, but eventually it will get there. But how? It's going to sort of go up pretty rapidly, and then it's going to sort of tail off and eventually get to fixation. If the alleles were codominant, the slope would look like this. Nice straight line, right? But with the situation of dominance, you get this curve. It goes up really quickly, but then it starts to tail off. Why doesn't it just go there? Why does it do this number?
Something with the proportions of the population, I mean, you just got.
Is it a heterozygote who keep bringing that small other allele?
Yeah. Where does A2 sort of, where can it hide? Right? So this one's bad, and you're going to get rid of genotypes that are this, but A2 can hide in the heterozygote, right? There's no fitness consequence in the heterozygote for having A2, because it's not codominant, it's completely dominant. Right? So it does change very quickly as you sort of diminish the homozygotes of A2, A2, but then as you sort of get down to the sort of lower frequencies, it hides in the heterozygote a little bit, but it can't hide forever, because the heterozygotes don't breed true, right? Eventually it sort of gets kicked out. Does you see that? So the rate of change is very quick at first and then slows down. If we sort of look at the average fitness of the population as we go across the range of frequencies of A1, and 1 being the best and 0 being the worst, as the frequency of A1 approaches 1, and as it gets to 1, then the population's average fitness selection is always going to increase the average fitness of the population. What is the most fit population in this scenario? given this set? What should the sort of homozygous A1 homozygotes only? When those are the only individuals in the population, that population is the most fit, given that scenario. Yes?
Yep.
Do you understand? Even though the heterozygote, but heterozygote's still got A2 in it, so it's going to occasionally breed out these A2 homozygotes, which is going to decrease the average fitness of the population, and selection is going to go, no, you're out, you're out, you're out. The selection, what's it do? Increase the average fitness of the population to its maximum, even at the expense of getting rid of all genetic variation, and it has nothing left to do. Does selection have any forethought?
No.
It's only seeking to sort of increase the average fitness of the population, right? Until it gets to the point where it has nothing left to select. So it doesn't have a brain, but it is trying to get here to its maximum average fitness of the population. And given this scenario, the maximum average fitness of the population is having a frequency of A1 of 1.0, meaning every individual is an A1 homozygote. Yes? So let's change the selection around to the selection on the dominant. A1 is still dominant to A2 in this scenario, but I've changed it around. Remember, dominance and recessive say nothing about fitness. It's a relationship. So here, the selection coefficient is actually showing that the dominant allele is the one being selected against. So the A1 homozygotes and the A2 heterozygote are now sort of less fit than the A2 recessive homozygote. Everybody see that?
Yep.
You good? I'm going to make the same kind of graph. I'm going to change it up a bit, so go ahead and you can redraw. I'm going to start A1 up at the top and A2 down at the bottom. So, what's the ultimate fate, or what genotype should be left, or genotypes, if you will? What genotype should be left after selection has sort of gone to its A2 should be the only one left, right? Given that scenario, the only genotype that should be left, which means the only allele that should be left in the population is A2. Gone to fixation, and A1 to extinction. Now, the question is not sort of, the endpoint is going to be the same. But how did it get there? Because A2, which is now started down here in the corner, is sure going to end up back up here. But in this case, because of the relationship over there of recessive and dominance, it's going to take a little longer. It's going to sort of do that. It takes a while to get started. But eventually goes to fixation. Unlike when in this situation, remember it jumped up real quick and then slowed down. This is going to sort of take a while to get started. It's going to take more of a sigmoidal and then exponential growth, exponential change, and then asymptote again. The difference when you sort of look at the change in average fitness is it takes a long time, but it still comes around and goes to the same point. The end point is the same. The pathway in which they get there is different. So this is with selection on the dominant, this is with selection on the recessive, and if you just did a codominant situation, it would be a very straight line right to it. So the endpoint is going to be the same. Selection acts to increase the average fitness of the population regardless. But the pathway in which the population takes is going to be dependent on the relationship between the alleles. Yes? No?
Yeah.
Yes?
Which one did you say? Which
Oh,
line
okay.
is it?
This would be the pathway of codominance, selection on the recessive, and selection on the dominant. The endpoint's all the same. Same for all of us, right? The endpoint's the same, but the path we take is different.
So for the recessive, it takes longer. But why? I don't see how it could take longer. They're both homozygotes, right?
Well, this is starting at a very low frequency,
Okay.
so the actual number of homozygotes, the actual number of most fit genotypes in the population is very low to start, but it's going to increase slowly, and then it gets to a point where it's sort of, then it becomes the most frequent allele, and it changes over, so there's a break point where once it crosses this sort of 50-50 line, and it accelerates it, and then it goes the other way.
What was the other one? It was not basically the same thing. What's the difference between the first and the second? They both have.
It's a dominant trait in the first one as opposed to.
Well, in both cases, A1 is dominant A2, in both cases. But in this case, A1 is the most fit allele, in this case, A2 is the most fit.
Okay.
And that's where you're illustrating dominant isn't always.
No.
Yeah, dominant doesn't mean most fit,
Yeah,
it's just, that's the trait there.
it's wild type.
Wild type. Who said wild type's the most fit?
No, I said that's what dominant would be, right?
What am I gonna do? Can I get rid of this? All right. If you look over on our little scorecard over there, we've done dominance and recessive and selection on both of them, but we still need to do, we've done codominance, so we still need to do overdominance and underdominance. I know you're excited, right? You are. Yeah. Here we are again. Candidates. In the case of overdominance, the heterozygote is the most fit genotype, right? So we give it a 1. It gets the 1. The homozygotes are not as fit as the heterozygote, so they get a selection coefficient, but they don't have to have the same one, so we use 2 different letters. So little s is a selection coefficient that defines sort of how much less fit the A1 homozygote is. Little t defines how much less fit the A2 homozygote is. Got it? No big deal.
How do you know which one's which?
Hmm?
How do you know which one's which? More or
The
less?
S or T,
Yeah.
you define it. As long as you define it the same way, you're all right. So, in order for the population to have heterozygotes, we need to maintain 2 alleles. In this case, selection is actually going to be acting, but allele frequencies would not be changing. Damn. Right? Didn't I tell you that selection would change allele frequencies? In this case, it doesn't. Because in order to have the most fit population, you need to have heterozygotes, right? And the sort of best proportion of heterozygotes is dependent on that little s and little t, how much different the homozygotes are from the heterozygote with respect to fitness. So we can calculate an equilibrium value. And in this case, we'll call it a stable equilibrium value. P hat. P hat's our equilibrium value, it's a stable equilibrium value, and it's calculated by a fairly simple equation, which is T, that little t over there, over S plus T. So, if we just sort of had an example of little t equaling 0.6 and little s equaling 0.4, in other words, the selection coefficients on the 2 homozygotes, then our p hat would be 0.6. In other words, our stable allele frequency that maximizes the average fitness population. This stable equilibrium value is going to sort of maximize average fitness. Remember that the average fitness of the population, that's what selection is going to try to increase. And so, in this case though, the heterozygote is the most fit genotype, so selection is going to try to keep both alleles around because that's the best scenario. If we look at sort of average fitness across a range of allele frequencies. So on the y-axis is going to be average fitness. On the x-axis is going to be a range of allele frequencies. Yes?
Is it just convenient that the S and T happen to equal 1, or are they always going to equal 1?
Oh, just that was totally my mistake. Sorry, don't ever think that. S and T are just coefficients that are independent of that. By chance alone. So, what's our best value for P that will maximize fitness? 0.6. This was about right here, right? Yeah, fitness of 1, so that'll be right around here. And anywhere close to where that might be. So when the population fitness is at its peak, the allele frequency of P will be 0.6, right? That's what that means. Which means Q will be 0.4. Because that's going to maximize the average fitness of the population. That's what that whole thing is trying to do. And what happens to fitness as we sort of move away from 0.6 either way? No, it's going to go down. So if the allele frequency changes in the population by some means, what's selection going to do? You're going to push it back. That's what selection does, right? If the allele frequency starts to wander this way, selection's going to say, get back there. Right? Selection's always going to be sort of pushing it to that peak.
You say when the population is at the 0.6 peak, it does what?
That's the maximum average fitness,
Okay.
right? So that's what that equilibrium value represents. When the population will be at its maximum average fitness, because that's what selection's doing. I've said it, right? That's what the whole thing's about. Selection increases average fitness. You got that? I have one more scenario for you, and since apparently I talked much slower today than I did yesterday, we're going to end after that. Okay? Ready? You don't have to have two. Or are you disappointed we're ending earlier? I will talk a little bit about something else, but just to introduce it so we can, next class, go right to it. So here's the situation of underdominance, our last in our hit parade of dominance relationships.
What's the plus?
Should be 1 minus.
Coefficient is.
Yeah. The plus makes the heterozygote the least fit.
Minus.
Plus means heterozygote is least.
When you add these coefficients, it's going to go up. Keep in mind though, depending on which textbook you look at for coefficients, sometimes they have 1 minus s, sometimes 1 plus s, and then they modify this to be negatives or positives, but it doesn't really matter. As long as the heterozygote becomes, if the heterozygote's the lowest one, you're dealing with underdominance. And in my little scenario here, 1 is the fitness of the heterozygote, and then these selection coefficients, which are defined now as 0.6 and 0.4, so this actually becomes a 1.4, this becomes 1.6, so the heterozygote is the least fit. Get it? So, the problem with underdominance is it produces what we call an unstable equilibrium. Of course. Just another turn of the screw. Have you ever read that story? It's like the scariest story ever. Like Stephen King. He knows nothing about the feeder. Actually, he does. But Turn of the Screw, written by Henry James, and that sort of old language. It takes forever. If you want to fall asleep, you can. But it is the freakiest story I've ever read. I just got chills thinking about it. So here's another Turn of the Screw. You didn't read it? Has anybody ever read it? Ever heard of it?
We're all illiterate.
All right. Try it sometime. It's not that long of a book. It is a little bit boring. Henry James, you really have to stick with him. But there's a big payoff at the end where you'll be able to give yourself chills any time by thinking about it.
Awesome.
I get the same feeling from, do you know The Shining, the movie? See The Shining? When those two little girls show up when he's riding the big wheel?
Like that.
The whole story's not that scary, but that's freaky. So this unstable equilibrium. You calculate it exactly the same way. You use little t and little s, and you end up with what? Oh, exactly the same answer. Lo and behold, because I like to do that to examples. So is everybody with me so far?
Yep.
So this does not represent the highest fitness. It actually represents the lowest. So when the population is at that unstable equilibrium point, the average fitness will be down here in the gutter. What it means is that's where the heterozygotes are really dragging the population down, right? Because they're the least fit, and you want to get rid of them.
Right.
So what happens to fitness if you go to the right here? You go this way.
Goes down.
Down?
What goes up?
Okay, go up.
Okay.
Let's see if it goes up to like there. What happens if you go to the left?
Up.
Okay, let's say like it'll go up to like there. Which way is the population going to move?
Right.
Left, right?
It depends on
Yeah,
where you start.
Yeah, well, let's say you start at 0.6, right in the middle.
I don't know.
No way.
I don't know.
Think of it this way. Can selection see which peak is higher there?
No.
So what's it trying to do? Increase average fitness. So which way is it going to go? Either way. It's unstable. Doesn't know. It can go either way. No matter which way the allele frequencies change, the average fitness is going to increase, which is exactly what selection wants it to happen. And then, what's it going to do? Is it going to say, is it going to get up here and go, wait a minute, I chose the wrong way? It's just going to go that way to its end. That's what selection does. So it's not going to decide like, hey, that looks better, I'm going to go that way. Remember, it doesn't have any foresight. It's just going to increase average fitness.
Is it possible for some of the population to go one way and
So
maybe if the population splits up or something?
another way to think about this number, this 0.6 or 60%, if you did this 100 times, 60% of the populations would go that way, and 40% would go that way. So underdominance has this, when the heterozygotes least fit, a selection doesn't care which way it goes, it's just trying to get rid of heterozygotes. So it's going to go for one allele or the other, it doesn't care. Because either way, but as soon as it starts down this path, it's not going to sort of make a U-turn and go, no, I should have gone the other way. No, it's going to do that. It's just going to continue, right? Because it's going to increase average fitness until it gets to its end. That's all it does, every time. That is the deterministic models of selection, and so
If 60% is going to go to the right, why? doesn't
why?
it go
Because
to the
P
left?
is 0.6, and that means that 60% of the time P will go to fixation, which means it'll go to 1. Remember, this is fixation. And we're looking at P. So 60% of the time it'll go to fixation for P, 40% of the time it'll go to fixation for Q. In other words, Q will become a frequency of 1.
Isn't fixation also on the top left?
No, that's average fitness to maximized. Do you have any questions on that? So next time we'll talk about, so this is what we were talking about, selection, adaptive evolution. Selection increases the average fitness and all about populations becoming adapted to their environment. Next time we'll talk about the other sort of big mechanism of evolution, and that is genetic drift. That is solely the role of chance. So think about it this way. You could be the most fit, most well-adapted krill ever to set foot, no, swim on this planet. Set feet on this planet. Yet, you could still be eaten by a whale. And all that's for naught. Right?
Right.
The role of chance and selection are always going on simultaneously. What we're going to do is sort of talk about the role of chance and then sort of put them together. And sort of how does chance and selection sort of play out in different populations. Because genetic drift is the other mechanism. We call it non-adaptive evolution. Natural selection is adaptive evolution. Genetic drift, non-adaptive. Because it's just chance. Allele frequencies change by chance.
Right.
If you put 5 populations and sort of grew them up in 5 buckets, alleles frequencies would diverge, and they would diverge over time by chance alone. You don't need selection to have change. So that's what we'll talk about next time with fun simulations and graphics and videos.